Thank you very much for the kind introduction. It's actually great to be back here again this year to represent ProBio. And today I'm going to speak about the overview and outlook of conjugation technologies in AXC. So I'll first start with introducing the concept of AXC and various conjugation technologies, and followed by an overview of ProBio CMC platform capabilities to enable the AXC manufacturing. And finally, I'll provide some future perspectives in this rapidly evolving field. The AXC concept, or somebody call it antibody plus, is a rapidly evolving term over the past decade. And in this term, so antibody plays the role of specific targeting, and the X plays the role of high efficiency function. And basically, in this X, this could represent a variety of different modalities. For example, if this X could be a potent small molecule to make the conventional ADC, it could be the radioisotope to make the antibody radioisotope conjugates. It could also represent nucleic acids to make, such as ASO or siRNA, to make the antibody oligonucleotide conjugates. It can be the peptide, so to make such as GLP-1 drugs to make the antibody peptide conjugates. And finally, it could represent complex systems such as immune cells and lipid nanoparticles to construct CAR-T, CAR-NK, and targeted LNP therapeutics. So the main purpose of AXC design is to achieve targeted delivery of payload, expand therapeutic window, and reduce the toxicity associated with traditional chemotherapy. So over the past few decades, there has been a lot of evolution for AXC design. So starting with early years with the first generation utilizing animal-originated antibody with high toxicity. And over the years, there have been a lot of improvement in the linker payload as well as the antibody. And we are now at the third generation of AXC, which mostly utilize fully humanized antibody with more hydrophilic linker, more potent drugs, as well as a uniform distributed DAR, utilizing site-specific conjugation techniques. And in AXC design, there are three key components, namely the antibody, the linker, and payload. So for the antibody, it should have a desirable affinity with low immunogenicity. And for the payload, they must deliver intended therapeutic effects without excessive toxicity. And the linker connects the antibody to the drug and should be released at specific conditions at the target tumor or tissue environment. So last but not least, the conjugation technologies basically determines how these key components are being connected and will be the focus of discussion for today's talk. So here is an overview of various enabling conjugation technologies for AXC. So each of them has their own pros and cons. And for example, for the conventional lysin and cysteine-based conjugation, utilizing random conjugation, the key advantage is their simplicity in operation, as well as have the most FDA-approved and clinical tested ADCs in the track record. However, on the downside, these random conjugations will result in a variety of DAR distributions and generate very heterogeneous products and product quality. On the other hand, there have been emergent site-specific conjugation technologies that enable more precise control of the DAR. For example, through the engineered cysteines, cysteine rebridging, on natural amino acids through enzymatic conjugation and glycol-based conjugation. So the key advantage of all these site-specific conjugation techniques are more defined DAR or even uniform DAR and very high conversion rate, a more defined conjugation site, and homogeneous product and product quality. On the downside, there are a couple of challenges for each of these methods. For example, a lot of them will require genetic engineering in the target antibody, so that might impact the stability or immunogenicity of the target antibody. And a lot of process involves enzymes in the reaction, so that will bring in some complexity and increase the cost of the overall manufacturing process. So in the next couple of slides, I'm going to do a little bit deep dive on each of these conjugation technologies. So starting with lysine cysteine-based random conjugation, basically utilizing the native residues in the antibody. So for the lysine-based conjugation, through amine coupling of the lysine site, since there are about 40 to 80 native residue lysine sites available in the antibody that is able to be conjugated, the resulting products will be highly heterogeneous. On the other hand, for cysteine-based conjugation, it basically utilizes the 4 pairs of interchain disulfide bonds in the antibody. So after the reduction of the disulfide bonds and expose the thiol groups, it could be directly covalently bonded to maleimide groups through microaddition reaction. In this way, the DAR value could be a little bit better controlled from DAR 2, 4, 6, and 8. However, the distribution of the DAR cannot really be precisely controlled given the nature of the random conjugation reaction. So utilizing similar cysteine-based chemistry, conjugation chemistry, we can introduce engineered cysteines at specific or desirable positions in the antibody to realize site-specific conjugations. The most well-known platform for cysteine engineering is the ThioMab platform, which was developed by Genentech almost 2 decades ago. So there are 3 key steps involved in this conjugation process. The first step is a reduction step, basically utilizing a very strong reductant to disrupt all the interchain disulfide bonds, as well as uncap the engineered cysteine sites. The second step after the buffer exchange is the oxidation step, basically is to introduce a mild oxidant to reform all the interchain disulfide bonds in the antibody. And the last step is the conjugation of the linker payload to the engineered cysteine site. So in this way, we can achieve precisely controlled DAR value and homogeneous conjugation sites. And at the same time, the stability of the antibody is improved since all the interchain disulfide bonds is being retained in this way. For the unnatural amino acids conjugation platform, basically the idea is achieved through expanding the genetic coding to allow the expression of unnatural amino acids in the antibody, which contains specific reactive groups such as ketones, alkanes, or azide. After the introduction of these unnatural amino acid residues, a direct coupling reaction with linker payload could be done through bioorthogonal reactions. So the most well-known platform in this area is Sutron BioPharma's platform, where they has established their cell-free expression system, producing the antibodies that contain the unnatural amino acids. Move on to the peptide-based technologies. This is also highly specific, and then basically this can be realized by introducing a specific recognition sites sequence in the antibody, and which could be specifically recognized by a particular enzyme. And on the other hand, also a second catalytic site should be introduced in the linker payloads end. And once both criteria are met, these enzymes could catalyze this site-specific conjugation with very high activity and specificity. And there are a wide range of different enzymes that could catalyze these reactions, with some examples listed here, such as Saltase A, MTG, FDase, and FGE. And lastly, there have been a lot of cases utilizing glycosyl-transferase technologies for site-specific conjugations. So the basic idea is to leverage the natural glycan residue structures in the FC fragment of the IgG, and utilizing these enzymes to introduce some engineered sugar residues that contains the chemical reactive groups such as azide, and subsequently through click chemistry to directly link the linker payload to these specific sites. There have been a couple of different platforms utilizing glycotransferase-based technologies, with the most advanced one from Synaffix, which is called GlycoConnect. And there have been a couple of ADC drugs currently in the late phase clinical studies. After an overview of this site-specific technology space, let's take a closer look at our platform capabilities to enable some of this AXC process. So starting with overview of the conjugation process flow, it involves first the production of naked antibody, or we can call it DS intermediate, which is not much different from conventional MABs or bispecific or other antibody format production. And second part is pretty much the preparation of the linker payload, followed by the conjugation production. And generally, with respect to the conjugation process, there could be multiple steps involved, such as the reduction, buffer exchange, oxidation, buffer exchange as needed, conjugation, post-purification, buffer exchange, and formulation step. So I'll just take two platforms as examples. First, the cysteine-based conjugation platform. And as I said, so this approach basically leverage the different reduction potentials in the 4 pairs of disulfide bonds in the IgG. So for example, for IgG1, typically the disulfide bonds between the light chain and heavy chain is a little bit easier to be reduced than the hinge regions between the 2 heavy chains. And we can fine-tune the reduction condition to get the average DAR as targeted. So there are a couple of key considerations in the cysteine-based conjugation process, starting first with the selection of reductant. So normally we'll prefer to use a strong reductant, and compare with DTT, the TSAP is more stable with a wide range under different pH conditions and the environment, therefore, and end up with higher reaction efficiency and does not react with a coupling reagent, and therefore is selected as a platform reductant. And also, can we control the overall reduction through reaction time? Not really the case. From this data, we can see at different reductant to MAB ratio and different reaction conditions, all the reactions turns out to be very fast and reach equilibrium within 1 to 2 hours. And considering the manufacturing operation time, it is really difficult to control the DAR value, average DAR value, by controlling the reaction time. And it's actually recommended and suggested to control the overall DAR value through the reduction ratio. And for example, theoretically, if we have 1 mole of TSAP versus MAB ratio, it can break down 1 pair of disulfide bond and result in an average DAR value of 2. And this should be linearly scale up as we increase the ratio of the reductant to the MAB. And which through the experimental results utilizing the model IgG1 molecule, the experimental data agrees pretty well with theoretical prediction, where we can easily achieve the average DAR 2, 4, 6, and 8. However, the DAR distribution cannot really be controlled, and most of this just indicate average DAR and result in the distribution of different DAR values. And taking TheoMap as a second examples, there are a couple of key lessons learned from our development. And first is the point mutations in the IgG plays a key role determining the stability of the mutated antibody. For example, in this particular case, we introduced point mutation and cysteine, engineered cysteine sites at 3 different spots in the IgG, and turns out to have a very significant impact on the purity of the IgG molecule. And just a recap for the TheoMap, there are 3 key steps: the reduction, fully reduction, reoxidation, and conjugation. And actually, the reoxidation is a key step through all this process to make sure that we can fully close all the free interchain cell bonds and allows the conjugation happen specifically at the engineered cysteine site. So it turns out to be the molar ratio of the oxidant to the antibody is the most significant factor determining DAR, basically to make sure as we increase this oxidant ratio, we are essentially making sure that all the free interchain cysteine is being closed. At the same time, the oxidant ratio and also the mutation sites on the antibody, as well as many other factors will impact, such as the antibody concentration, etc., will impact the stability and purity of the conjugated products. Therefore, all these factors need to be carefully balanced and optimized in order to generate a fully optimized conjugation process. And here is an example of optimized process for a model IgG molecule for thiomab conjugation. As you can see, after optimization, basically all the interchain disulfide bonds should be well oxidized with very minimal DAR value generated from these free thiol groups from the interchain disulfide bond. And essentially, all the conjugation should essentially happen at the engineered thiol group. And the resulted DAR is highly uniform at the target DAR2. And finally, I'd like to speak about a couple of key trends and perspectives for the future of AXC field. First, one of the key observations we have, as we can see over the past decade, especially from the recent years, we can see that ADC or AXC are rapidly evolving from random conjugation to site-specific conjugations for improved homogeneity, stability, and manufacturability. In particular, starting about 2-3 years ago, essentially all of the ADCs currently newly entering the pipeline utilize site-specific conjugations rather than the conventional random conjugations. And if we do a deep dive into the site-specific technologies being utilized, we can realize that the majority of them use enzymatic-based conjugation or glycol-based site-specific conjugations. So starting last year, we also established our R&D platforms to support this emerging enzymatic site-specific conjugation applications. For example, for these particular case studies, we utilize MTG for site-specific conjugation. And basically, this recognize the Q295 spot in the IgG. And also at the same time, we introduce the N297A mutation to remove the glycosylation and enhance the coupling efficiency at the desirable site. And after the introduction of this linker that contains the IZI groups, the for subsequent conjugation of different linker payload could be realized simply through click chemistry reaction with the IZI. And as you can see, the resulted DAR is highly uniform at DAR2 and with pretty high SEC purity and can be easily scaled up at in the lab scale process. And additionally, we have developed a single hydrophobic chromatography step to efficiently remove the excessive enzymes, MTGs, and excessive unreacted linkers through a one-step chromatography step purification. And the second key trend is increased utilization of antibody fragment in the conjugation applications. So we have worked with clients in different AXC fields, such as the RDC, the AOC antibody nucleotide conjugates, and quite a number of cases for in vivo CAR-T applications. And given the very small size of the antibody fragment, as well as their simple structure, there are a number of key advantages of using antibody fragments in these applications. For example, their superior tissue penetration and delivery. It's very easable and flexible in engineering and allows conjugation. For example, you can easily realize site-specific conjugation by introducing cysteines at the C-terminal of these antibody fragments to realize specific conjugation. And also improved safety and drug-like properties. And there have been a number of different types of fragments, such as utilizing these applications, such as Fab, single-chain FV, VHH or nanobody or Fab2 fragment. So it's worth mentioning that ProBio has also established our CMC platform process for these various different type of antibody fragments for VHH, SCFV, and Fab. So for the cell line development, which only takes roughly 10 weeks from DNA synthesis to top 6 clone production, and the titer of expression could be up to 15 grams per liter. And we have established our dedicated downstream purification and analytical platforms for these different antibody fragments to help accelerate the speed of and quality of the overall program. And we adopt light CMC strategies for these antibody conjugates programs, and particularly for nanobody like VHH, we can take as fast as 3.5 months from DNA synthesis to IIT material production, and as fast as 4 months from DNA synthesis to tox batch production for IND applications. And we offer flexible GMP production scale from less than 50 liters up to 2,000 liter scale. And I think there should be 11, but anyway, this so far over the past few years, we have generated over 11 antibody fragment CMC projects, and with the most advanced VHH projects has received IND applications from the US, Australia, and China. With that, I would like to thank everybody for your time, and happy to take any questions you may have. Thank you.
All right, thank you, Charlotte, and thank you, everybody, for being here today. Hopefully we get that sorted. So, yeah, we're in a golden age of molecular and increasingly cell engineering now. You know, we can create things that we never dreamed were possible, to do things that we never dreamed were possible, even as recently as 20 years ago. We're stitching together biological molecules and other components in ways that we never thought of before, and in ways that are totally unnatural. We're building Frankenstein molecules. Now, there's good reasons for doing that, but there's also an awful lot of complexity that brings that we need to think about right across the development cycle. So my objective today is to sort of bring all the technology we've just heard about in the last two presentations into a framework for those of us who are developing products to be thinking about how do we wrestle Frankenstein. So what are we talking about? The platform we lived with up until about 20 years ago was built on small molecules and monoclonal antibodies. Small molecules, pure chemical species, easy to completely characterise. We'd known about them. We'd been dealing with them as drugs for almost 100 years or longer. Monoclonal antibodies were relatively new, but they gave us a single target to address, and they started to introduce a few concepts around complexity because no longer did we have a single molecular entity in our product. And so for the first time, the regulators had to deal with the concept of variability. I spent 3 years of my life negotiating biosimilar pathways with the European Medicines Agency, thinking about how do we do comparability between a non-glycosylated protein called filgrastim and a biosimilar version of it. That was pretty simple. EPO was a little bit harder because of its glycosylation profiles. And ultimately, we ran down an argument that Big Pharma couldn't control their own processes, so how the hell could we as biosimilar developers do it? That was our first inkling of complexity. Now think about what we've got now. Fragment antibodies became the next step. We heard a little bit about those. You know, there's a reason why they don't exist very often in nature, is because all the stabilising elements aren't there. Then we started doing bispecific, and these days we're talking trispecific and even tetraspecific antibodies. I think, I can't remember who was it, Ying or Charlotte spoke about it, but you change one amino acid in these things and you totally change yield, aggregation propensity, stability, a whole bunch of things that make a pharmaceutical product. And then we started putting non-amino acid things on the side of these molecules to produce RDCs, ADCs, AOCs. And these days we're even doing it with whole engineered cells when you get into the cellular immunotherapy space. So the common theme here about these new modalities is this increasing amount of complexity as we put more and more pieces together. Now, why the hell are we doing this? And why does it matter? Well, the answer is we're trying to solve problems that traditional therapeutics couldn't. If we pick the cancer examples, you know, tumour heterogeneity and resistance. Tumours don't express a single set of antigens. So a single antibody targeting a single cell surface receptor or tumour-associated antigen isn't going to get rid of all the tumour. There's plenty of targets that antibody drugs can't hit, intracellular proteins being the most obvious one. going back to cancer again, immunosuppressive microenvironments that actually keep our immune cells out. So you can get an antibody in there, but there's no immune cells there to recruit. So we created bispecifics to actually attach to the T cell and drag it into the tumour, right? Something we couldn't have done possibly before to overcome a real disease-based problem. And then we have the whole question around precision delivery. I mean, there's a reason why we do ADCs and RDCs is because the payload is toxic. And so we want to deliver it in as a focused way as possible without the risks of systemic toxicity. So these are the kind of problems that our ingenuity is trying to solve today. And, the outcomes are starting to emerge. We've got 15-odd ADCs approved, 15-odd bispecifics approved with another probably 15 or so pending. There's 4 RDCs approved now, and I'm not counting the diagnostics here. This is the theranostic versions. We've got 11 cellular immunotherapies approved and over 40 cell and gene therapy products approved now. The size of these markets by 2035 is going to be significant, but remember the scale on this chart is a log scale, by the way. Right, so small molecules still dominate, will continue to dominate, as will monoclonal antibodies. If you can, drug developers out there, if you can hit your target with a small molecule, do it. If you can't hit it with a mAb, only then do you think about these more complex modalities. And that is why small molecules and mAbs will continue to dominate, and these complicated modalities are going to go to the spaces where we haven't been able to solve them today. I thought this was interesting. I actually asked Claude to produce this, where are all these technologies sitting on the Gartner curve? And I had a few issues with them, and I've moved a few of the dots around, and anyone else who was looking at this will probably do likewise, right? So you can have a debate. But we have to accept that these technologies are going through cycles. And that flows through not just the ability of technology to deliver, but also it flows through with the ability to fund these types of programs. Capital flows onto the front end of the hype curve. It then evaporates when you're in the trough and might come back again once you get into the sort of productivity space. Okay, so what does that mean for us as a drug developer? We need to think about this across, and I've thought about this across preclinical and clinical development, about manufacturing, and then from a regulatory perspective. So from a preclinical and clinical development perspective, out the gate is the question of characterization. It's already been mentioned at least once today. We're not talking about a singular molecular entity now, although if you can get there, that's great. And so all the ideas around precision conjugation are really, really important. Not just because you've got better control of your molecule, because it makes it so much easier to then satisfy a regulator that you can characterize your product. It also makes it easier for you to move your production process from one site to another, because little things can change your DAR, can change the ratios of different components in your bispecific antibody, because at the end of the day, it can conjugate in all sorts of different ways. And that's really important because you have unintended consequences from some of those changes in distributions. We've got mechanism-based toxicity to deal with. Cytokine release syndrome and ICANS with CAR T-cell therapies, for example, or bispecific T-cell engagers. You're trying to drag your immune system into doing things it wasn't doing before. Well, it can overshoot pretty easily. So the therapeutic windows of some of these drugs can be really, really tight, something we need to get on top of very early. Pharmacology, dose-response curves are out the window. In CAR T-cell therapy, for example, there isn't a linear dose-response curve because you get such massive in vivo expansion once you administer the product. Similar things happen with radioconjugates, particularly as you're probing that narrow therapeutic window that you might have. You've got multi-specific antibodies, you've got avidity effects happening. You've got bystander effects happening that you can't necessarily predict and are going to be dose-dependent in ways that we don't necessarily fully understand. And increasingly, we're going to need companion diagnostics to go with these molecules because A, the payers want to know that you're treating only those patients who are going to respond. And B, if you're targeting antigen A and T-cell receptor B, then you better make sure the patient's got both of those. Otherwise, the regulators are not even going to approve you, and your clinicians aren't going to bother treating a patient who doesn't. And so not all of our companion diagnostics are available off the shelf today. So that's a starting point for the preclinical and clinical things we need to be thinking about, and we need to think about these obviously as early as possible. Then we go into manufacturing, bispecifics and ADCs we've touched on already. You can mispair the components. You can get different ratios of the different components. We heard slight changes in your oxidation conditions are going to result in changes in your DAR ratios. You've got to have control of those, and you've got to understand, and the regulator is going to insist that you understand how sensitive is your process to those. How much in-process control do you have to have over those oxidation potentials, the duration of oxidative or reductive conditions you're imposing your molecule to. Supply chains are becoming a bit of a nightmare. Radiotherapeutic drug conjugates, for example, your isotope has got a short half-life. You're making just in time. Autologous cell therapies are the same. You're making patient-specific just in time. That creates masses of complexity on the clinical sites. The list price of a CAR T-cell therapy today is about $500,000. The total cost of administering the CAR T therapy is $1 million, because there's another $500,000 of clinical costs associated with delivery. That's a big deal when you think about affordability and payers. Something you need to take into account and design your product to minimise that impact on the clinical setting. And so the message, and this has been rammed home to me by mistakes, by all sorts of things, by being a CDMO along the way as well, manufacturability must be part of your target product profile. And by manufacturability, I'm talking total cost of ownership. I'm not talking bulk synthesis of the drug substance. It's everything along the chain from tissue collection, isotope creation, through to delivery of the vial to the patient and what setting that's going to be required to be delivered in. And so all that comes together with your regulatory and commercial pieces. These are novel technologies, and there is no regulatory pathway or accepted regulatory science for many of these. It's emerging. The regulators are learning rapidly. They love talking to novel developers because they actually learn as much as we do from the interaction. But there is no single framework, and the regulators are creating novel pathways for each of them. Regulators will always tell you engage early. I'm sure we'll hear that from Madhvi early, but how do you do that? And what do you do that around? And what do you need to have for that? And how do you frame your questions around that? And what knowledge are you bringing to the table to facilitate a useful conversation? The regulators aren't going to teach you how to do the job, but they will engage you in terms of how to manage a new technology through the system. Many of these therapies, particularly the cell therapies now, are becoming potentially curative with a single dose. That totally disrupts the way payment systems work. And we're still wrestling with that now. And I've already touched on the fragility or lack of robustness of your supply chain. So thinking through all of those are going to be critical to your commercial success. In cell and gene therapies today and radiotherapeutics as well, CMC and manufacturability is the number one question that big pharma ask when they're looking at licensing a product. They'll take your initial clinical data for granted. If it looks good enough, it's all about CMC. So if you haven't factored that in, and many of us haven't traditionally in our development when we're coming out of the lab, then we're not going to get anywhere and this product won't make it to market. So is it worth it? Well, yeah, it is, because we're solving problems that nothing else is able to solve. But let's make sure that's what we're doing. Design your manufacturability from day one, not after you've done your clinical win, because it's too hard. You're locked in at that point. The comparability challenges are significant. I'm a big believer in the idea that the product is not the process. We must get away from that from a regulatory science point of view, but you can only do that with a data set that says you understand your product and you can control it. Engage early, calibrate to the modality. By that I mean make sure that you understand where the modality and the science in that modality is going, where it's going to get used. Don't be a technology looking for a problem to solve. Be solving a specific problem. And it needs so many more disciplines to come together more early in the program than we had before. So yes, it's worth it. Yes, it's complicated. We need to keep wrestling Frankenstein because the patients out there that have no therapies today are relying on us to do it. Thank you.
I might start with Madhvi. Thank you very much for being here from the TGA. Thank you. I might start with, for developers working on bispecific antibodies, what are the key regulatory considerations they should be thinking about early, particularly before moving toward clinical development or registration?
Hello, can you hear me?
Yeah, thank you.
Sure. So when I would say, first of all, the most important thing when you're thinking about a clinical trial moving on for registration would be familiarizing yourself with the international guidelines. Because everything, all regulatory agencies do need compliance with the international guidelines. So, such as the ICH, I think most of you would be familiar with the International Conference for Harmonization of Pharmaceuticals Used in Human Studies. So the ICH guidelines, the TGA has adopted the ICH guidelines as well. And so the ICH guidelines do prescribe and guide you for all the manufacturing processes, testing, etc. They're different guidelines. So I would say at that stage, you start adopting and using those guidelines to plan your manufacturing process, your assay systems, etc. The other key, and I'll come back more to it, the other key criteria is making sure that your manufacturing practice complies with the good manufacturing practice. So this is important for clinical trials, and then it's a step further. When you come for registration, there is a requirement that all manufacturing process has to be conducted on a GMP certified or licensed site. So you actually need a site that has GMP clearance or that has a license. So those would be the first of all the two most important things. Now coming to the more technical things. Obviously, when you are manufacturing, you would need detailed characterization of your drug substance, drug product. You would also be thinking about raw material validation. So also thinking about testing your cell lines or the master cell banks and working cell banks to test for any adventitious agents, because there are regulatory requirements, and then again there are ICH guidelines and there are pharmacopoeias that will tell you how to do it and what is expected. Again, the assay systems that are used for characterization and for testing should be validated. The other important thing is also stability. So when we think of stability, generally the impression is that, yes, you need stability just to test for or assign a shelf life. That is true for drug substance and drug product. However, it is also important that you have data for your intermediate product, which, especially to support the hold times for the intermediate product. The other thing is, again, for stability, there is prescriptive guidance on what is required, how many batches, how many samples. And you have to start thinking about it much earlier, because for the initial registration, we do consider the specifications and that you set up in your pivotal clinical trial data. So, you start thinking then. For biospecific antibodies, which is the subject matter today, I would say think about assay systems, quantitative assay systems, not just and qualitative, but more importantly, quantitative. And the reason I'm emphasizing that is we all know the issues, the main issues with the biospecific antibodies. We heard today, Tim mentioned about purity, about, mismatched products, fragments, aggregates. We are all familiar with that. So purity is very important. And, all regulatory agencies, including the TGA, does need qualitative assessment of these impurities. For GMP, the other consideration would be if you're thinking of manufacturing it within Australia or overseas. So that will decide whether you need a clearance or a license. But the obtaining GMP license or certification, you would anyway need TGA's GMP clearance or GMP license. Either way, if it's a overseas manufacturing site, you'd need a clearance. If it's national manufacturing site, you would need a license, and that is a time-consuming process as well. So think about that, start planning. Something that we don't really think about is names. So please do start contact and talk to the WHO, because there are naming conventions. We can't just name our products whatever. So the names also have to comply with the INN, and also then the TGA also does a check on the names when people come to register, sponsors come to register products. Again, obviously, when you're talking about thinking about clinical trial or even registration, think about scalability. So we are talking about quality, and we've talked about purity. What I haven't touched on is non-clinical or toxicology data, and also clinical trial data. So think about stability, think about impurities, think about toxicity, and use assay systems that will clearly characterize everything. One thing I didn't mention, when I say characterization, we do look at sequencing as well. So all your, constructs and also the cell lines, we do look at, so your assay systems that you're using for identity, affinity, avidity, purity, everything has to be validated, and everything has to be specific for the product.
Thank you. Thanks very much, Manvi. I have another question for you. Are you happy to take another one? Thank you. For new manufacturers or sponsors, could you briefly outline the main pathways available to register a new medicine on the ARTG, and what factors might influence which pathway is appropriate?
Thank you. So I can tell you that we do have different lines or different streams of pathways of registering new medicines at the TGA, and I'm talking about the ARTG, which is the Australian Register of Therapeutic Goods. So the standard, you would mostly be familiar with the standard pathway, where it takes around 220 days after a submission is accepted for evaluation, and from evaluation to decision phase, it is 220 days. Then we also have a priority pathway, where you need to apply to the TGA, and the clinical delegate makes a decision whether or not an application or submission can be accepted within the priority pathway. The priority pathway, as is suggested by the name, is for medicines of unmet need, which are critical, and so it is a much concise or shorter. They're shorter time frames. We have to evaluate and do everything within 150 working days within the priority pathway. Then there's also the provisional approval pathway, and that is, again, for life-saving drugs or medicines. And the provisional pathway actually allows, sponsors who don't have full clinical trial data. So you might still be getting clinical trial data. It's not sufficient, but again, if they are critical medicines, then you could apply for the provisional pathway designation, and the application will be assessed whether or not it can be accepted based on clinical need within that provisional approval pathway. The provisional approval is only for 2 years, after which we do expect the remaining clinical trial data to be available. However, based on data and circumstances, there can be extension up to 2 times. The other pathway is the orphan designation. So that's for rare medicinal needs, or rare therapies. And in the orphan pathway, there's a fee waiver. So that is because it's for rare diseases. So, and you don't need to pay the evaluation fee or the application fee. The standard timelines apply, but then there's a fee waiver for the orphan drugs. We also have some international work share pathways. So we have the access pathway where we actually work with other regulators. So access, as the name suggests, it's between Australia, Canada, Health Canada, that is, Switzerland, and then Singapore, and MHRA, so UK. And so we actually share, so sponsors can actually submit just one dossier, and they, if they want registration in different countries all at the same time. So I'm not saying the decision will be at the same time, but you can submit your dossier, it will be evaluated at the same time, and then the decisions will be made independently by each jurisdiction. And so it may be staggered, but then the evaluation is done together, and we often share the modules. So it might be that the quality, which is within the module 3 of the dossier, may be evaluated in Australia, and then Singapore might be doing the toxicology or module 4, and another country, Switzerland might be doing the module 5, which is the clinical trial data. And then we all come together throughout the evaluations. We discuss with each other. There's only one set of questions that is sent to the sponsor. So it's a much more straightforward, easy for a sponsor or manufacturer who wants to register their medicine in different countries. The other pathway that we have is the core pathway, which is the comparable overseas regulator pathway. So there we actually accept evaluation reports from other countries, and we take them into consideration for our evaluation and decision making. So that is a much quicker, process. And usually we accept reports from the EMA and the FDA. The requirement, the main requirement is that the full dossier and full report has to be available from that overseas country. It can't be redacted. The other pathway that we also have is the Project Orbis pathway, and that is essentially for cancer medicines. And in the Project Orbis pathway, there are concurrent evaluations. So we have the access partners that I just mentioned, and there are a couple more countries within the Project Orbis pathway. So each country does their own evaluation, but they're done concurrently, and we do discuss with each other. So these are the different pathways, depending on how much clinical data is there and how urgently a medicine is required.
Thank you so much, Manvi. Thank you. I have a question for Dr. Lee. Dr. Lee, from your perspective, what are the key innovations driving transformative change in CMC development for complex biologics?
Yeah, thank you for the question. Innovation is always a very passionate topic for me. And so from my perspective, there are a couple of key areas driving innovations in the CMC space. And so first, and also these are the areas our company is currently heavily invested in. I think, first of all, is to establish a digitalized platform, basically incorporating some advanced tools such as AI functions in our digital system. To realize this, basically we need to set up first an IoT structure, connecting the lab instruments along with experimental workflow, the process knowledge, as well as all the process data into one centralized digital system. And after that, we can incorporate some AI models, such as large language models, and combining with some mechanistic models, such as based on first principles, to better understand and learn this historical data, which is tons of data, and hopefully utilizing these digital tools to better help us guide the future process development, do the troubleshooting, and do a lot of automation functions such as data analytics, generating reports, prepare a lot of presentations, etc. And so that is one of the key pathways to improve the overall efficiency and the throughput of future CMC programs, particularly for CDMOs. And second areas, I think very exciting is the cell line engineering. I think for every CMC program, a very good starting point is always generating a high producer cell lines that could generating your protein of interest with a desirable CQA profile, particularly for complex modalities. And with some advanced tools such as CRISPR-Cas9 based gene editing tools that give us more leverage and help us to basically do the genetic engineering more creatively to help design our host cells in a particular way with desirable feature and all express the proteins of interest in the particular feature. Another area on the analytics side, we've seen an increasing trend of utilizing inline PAT analytics, such as Raman-based technologies to allow in-time monitoring and in-time feedback control of the overall process. Basically, we can get a lot of in-depth data from both upstream and downstream to help us better understand all the different changes in the process and help us in the future to control the overall process in time and automatically. And last but not least, I think another topic I just speak today is on the antibody conjugates. Obviously, there are a lot of exciting innovations happens, particular around the improvement in the linker payload as well as the site-specific conjugation technologies to allow for higher conversion and higher purity products with more desirable quality features. And with that, I think there are a lot of other very exciting fields in the innovation, and I'm very excited to see what's happening in the future.
Thank you so much, Ying. And I have a question for you, Tim. How early should companies be thinking about the target product profile and manufacturability? And how can those decisions affect CMC, scale-up and regulatory strategy?
Well, out the gate, pretty much. So as soon as you move from being a fundamental research project to something that you think you want to turn into a product is the key point, right? What's that product going to look like? What format is it going to be? How's it going to be administered? What disease is it going to treat? What concentration am I going to need it to make it viable, right? If you're trying to do subcutaneous injection, for example, then it's got to be less than 1.5 ml because you can't inject any more than that. What's the price that I'm likely to get for the product? And therefore, what's my cost of goods going to have to look like? Because basically you take price, divide by 10, and that's the maximum cost of goods you're allowed for big pharma. Right? They want a 90% margin plus. Small molecule, they want 99. That's just the way the industry economics work. And so that influences whether you can tolerate a substandard yield or whether you're going to have to push your biological yield to the 10 or 15 grams per litre that's kind of state of the art today. If you've got a highly potent molecule, you can get away with a less efficient process. But at Adalta, our first product was a fragment antibody fusion protein. And we had a lot of conversations around and went into phase 1, very nervous around whether we were actually going to need, where our dose was likely to come out of phase 1 versus our cost of goods. Now, it turned out it was okay. But it also meant we had a continuous manufacturing improvement program in place from day one because we knew we were needing to stretch harder than our first product came out the door. Now, that has its own challenges because, and getting back to my theme around product as a process, if you don't understand your process very well, every time you make one of those process tweaks, the regulator is going to often start from a position that that's a new product. because you don't know all the downstream consequences of subtle changes in glycosylation profiles, in subtle changes in ratios of different components in your mixture of product on the safety and efficacy of the products. Unless you understand that and you've got that characterised really well, then even a continuous manufacturing improvement program isn't going to help you, because you could invalidate everything you've done before that. So that's why TPP is so important from the get-go.
Thanks so much, Tim. I might open up questions to the floor, please. Has anybody got any questions for our panel? Yes. Microphone coming to you now.
Thank you.
Thank you.
Thanks. Hi, Penny Field, Keystone Biologics Advisory. I have a question about the impact of the FDA guidance that came out in January in terms of flexibility of manufacturing in terms of cell and gene therapies, and how that's going to impact, or if you consider that this is going to have an important impact for you. I'm not too sure who wants to answer that. A number of people would answer that particular
Matt, do
question.
you want to start and then I'll have a crack?
Sure. So we do consider the FDA guidance. We usually, the TGA adopts ICH guidelines and EMA guidance. And these guidances are again reviewed, and then we consult and have stakeholder consultations, and then we adopt either with or without comments or some amendments or extra explanatory notes to it. So we have not yet adopted the FDA guidance, but definitely there will be, when there is a guidance out there, there is discussion, and we do start considering how it will impact. So I don't have an answer for you today, but obviously as guidances come out, they do impact internationally, because there will be discussion. There'll be discussion amongst the regulators, and then we will see how we can whether or not we adopt it, how we consider it is again it's independent, it's product specific.
So from an industry point of view, I'm really encouraged by what the FDA had to say in January. I guess it's important to acknowledge that it's FDA policy as opposed to guidance. And so what they're signalling, as they've done with several other areas, is a reflection on the advancing understanding of the manufacturing processes and the impact of manufacturing and outcomes in cell and gene therapy. In the early days, it very much was product is the process. And if you changed anything, you changed a flask, you changed a reagent, you changed anything, they were sending you back to the clinic, which means that some of the early CAR T products, and Kymriah and Yescarta are hamstrung by this today, couldn't make any changes whatsoever without the risk of invalidating all the clinical data and preclinical data they already had. Now, that world is now changing, and it makes a lot of sense because, on the one hand, we've got a highly variable product, particularly an autologous CAR T cell. The single biggest source of variability in a CAR T product is not the manufacturing process. It's the patient starting cells. So all of this stuff that we're doing in controlling our manufacturing process is a rounding error on the variability you get from the incoming apheresis product. And I think what the FDA has said is, yeah, we kind of get that. And so I think what's going to happen, and I'm encouraged that it's going to happen, is we're going to be able to start saying, I'm making this process change. Here's what I know about my process. Here's what I don't know about the activity of that product, by the way, because the other point is the variability of the product itself. You've got multiple clonal inserts, for example. It's not a monoclonal product. And we know from work that UPenn did that it can be a single clone is enough to result in an outcome for a patient. We just don't know which clone it is. And we don't know which one or which set of clones are coming out of each manufacturing process. So we do a lot of work around exhaustion markers and memory versus effective phenotypes and all that kind of stuff, but actually we don't know. So making those release criteria is dumb because you're potentially isolating or not having a patient do it. So broadening our release criteria, controlling the stuff that's going to bring adventitious agents in, doing risk assessments to say, where might I change the product? Right, I changed my vector. Well, I think you might have changed the product. Changing a source of a particular cytokine, probably not. But having said that, another process that I'm involved in, just a poor batch of flasks can influence how our cell expands, and you can get batch or you can't get batch. Now, if your process controls are okay, you can pick it up. Should that invalidate your manufacturing process because you've got to go back again and redo all the clinical? Probably not. But there's some of the reasons why product as a process is so important. But I'm encouraged with where the FDA is going.
Thank you, Tim. That's and thanks, Madby, as well. Thank you.